Thanks very much, Jean-Louis, for the kind introduction. Good morning, ladies and gentlemen. I think Frank and Olvia have set us an excellent example, and I hope that this presentation will reach the high standard that they've already set for us this morning. So we're going to continue the discussion with the second case study, which is from Pfizer, and it's focused this time on the development and verification of design space. I'll do the introduction and the first part of the presentation, and then I'll hand over to Tuna to carry on the presentation, and I think I'll do the last few slides as well. So to begin with, this was the team that was involved. Thank you very much to everybody on the team. Not everybody can be here today, but we did have a very rich series of discussions, and it was hard work, but it was very interesting and fun, and hopefully that will be reflected in what you'll see in the following slides. So we're going to start by looking at the case study itself, and we'll also give you some background on what I've called Product A, the product that was approved and is the subject of this case study, before we move into a series of discussion topics where we'll look at the development of design space, some discussion around the scale-up and verification aspects, and then also presentation of design space in the submission. And we'll present some best practices that we think may have come out of the conversation, and also some topics for discussion in the questions and answers afterwards, but more particularly probably in the session tomorrow. From a Pfizer perspective, Pfizer actually uses and applies quality by design concepts as a way of life now in the development of our new products. And we've got data that we've talked about publicly, which shows that from an industry perspective, we get a significant benefit in terms of improved robustness for our products and processes as compared to products that were developed using more traditional approaches some years ago. What we've seen, however, recently is that there's a significant effort in putting together some of these so-called QBD submissions. They take significantly greater resources on a company side. The files are often quite detailed. We've seen that already in the first case study. And the impression that we get is also that this can be challenging for assessors as well. So it takes more resources both to put these together and to review them. But there's a real value, I think, as Georges and Jean-Louis have indicated this morning, in terms of trying to present this enhanced knowledge in submissions. And therefore, the key question for us, I think, over these next 2 days is how can we do this in a way that is more optimal than where we are at the moment, and how can we use this information effectively? In particular, the focus of this presentation will be concerning the design space concept and how we can use this concept, which has been built into the European variations regulations, and get the value of this concept more effectively. Product A is actually a relatively simple immediate release film-coated tablet dosage form. It's indicated for advanced renal cell carcinoma. The tablets are made by a very conventional dry granulation manufacturing process. This is kind of a platform process that we use within Pfizer for pretty much all of our tablet products. And as far as the drug substance is concerned, as you'll see, it's a 6-stage synthesis process using 3 starting materials and includes a number of different chemical transformations, isolations, and crystallizations. During development, a number of different crystal forms were identified, and one of these, a single crystal form, which was actually non-hygroscopic, was selected for commercialization. Obviously, being non-hygroscopic, that helped from a stability perspective. So that's a bit of background about the case study and the product itself. Let's move now into the meat of the presentation and the case study with looking at the Pfizer approach to development of design space. So this is a pretty wordy slide, but in essence, it's how to do QBD on one slide, and there's nothing that I would argue is unusual or different from what you may have seen other companies do. So we would consider this, I think, a typical approach following the QBD principles that are laid out in the ICH documents. Understand what needs to be delivered to the patient through the quality target product profile. From that, derive the critical quality attributes for both the drug product and the drug substance. Understand what aspects of the input materials and the process build quality into the product, and how those links between the process parameters and the critical quality attributes actually operate in practice through a mixture of risk assessment and experimentation based on prior knowledge, and ultimately from that determine the manufacturing process that will deliver that quality. And the ranges could be expressed either perhaps as proven acceptable ranges or as a design space. So to try and use the example from this particular product, I'm going to talk mainly about the drug substance, but the principles actually that we'll talk about in the next few slides, in essence, are the same kind of approaches that we would also use for drug product. So obviously we use risk assessment to identify the CQAs, and for the drug substance, one of these was palladium, which was used as a catalyst in the process. And inevitably with a drug substance and a small molecule drug substance, there's a big focus on control of the impurities. One of the things that we've done in the submission is to include what we call impurity mapping grids, which show where impurities are formed and controlled in the process. And I've got actually an invented example, although it fooled a lot of people in Pfizer when we were reviewing this who thought it was real. But it's an invented example. And you can see that in essence we lay out the drug substance process across the top of the chart, and you can see in red where impurities are introduced at the different steps, and the green impurities show where they're controlled. So it's a very easy and nice visual summary of the drug substance process that helps from a sort of thinking about control strategy perspective. One of the things we do is we use a variety of different risk assessment approaches, and one of which is to actually rank the process steps themselves for their importance in delivering the different critical quality attributes of the drug substance. And we focus on what we call, or what you can think about as the control gates for the impurities, typically the isolations or the bond-forming steps. And from that, as you would imagine, the later steps in the process tend to be the highest ranked steps, and the earlier steps tend to be lower ranked because the impurities don't propagate through the process. But that's not always true. As you can see, one of the earlier steps was actually ranked quite highly because of the importance of that step in the origin and control of certain impurities in the process. As far as design space development is concerned, the design space, the Pfizer approach has been to develop a design space for each process step in turn, but then actually combine them to give a design space for the whole drug substance process. And the same kind of approach is done for the drug product. We determined the preliminary trends on the impact of process parameters on the critical quality attributes using a mixture of risk assessment based on prior knowledge and also often univariate experiments done early in development. One of the features of this and other drug substance processes that we've developed is that we try and understand the impact of the mixing in the drug substance manufacturing process using a variety of different equipment configurations, and in essence try and design the process such that mixing doesn't have an impact. So we're trying to take out one of the scale factors during the development of the drug substance process. When we've got this initial information and we use the risk assessment to identify the highest risk process parameters, these are then studied in more detail using multivariate design of experiments. And from that we can then derive models on which we can develop a design space, obviously based on those parameters which have the greatest influence on the critical quality attributes. Just to give what I've talked about a little bit more clarity and to make it real for you, I've pulled out one example from this which relates to the design space for the step 5 crystallization step, which actually consists of a polishing filtration from the output of step 4, followed by a deprotection of an acyl group and removal of the palladium catalyst. Then the crude drug substance is crystallized by addition of an antisolvent and then re-slurried and further purified in the final step in the process. The development scientists found that from preliminary experiments that a number of parameters that were considered actually didn't have an impact. And I've just given you a few examples there. But they then went and did a design of experiments to study these interactions or potential interactions of the different parameters. You can see a number of parameters were selected, the kind of parameters shown on the slide there, order of addition of the components, anti-solvent temperature and quantity, for example. A focus, we look at all the impurities, but obviously we're focused on the particular impurities that are associated with this process step. A couple cited there, as well as the palladium. And then obviously from the experimental work, a number of conclusions were drawn. The anti-solvent quantity was found to have the biggest effect, followed by the temperature. One of the impurities didn't seem to be impacted by any of the parameters investigated. And as was mentioned in the first case study, as well as looking at things from a quality perspective, we also have economic considerations that may be included in part of the experimental design. And in this particular case, we found that a couple of parameters affected yield, but not quality. So that helps us figure out where we want to operate from an economic perspective and also deliver quality drug substance. As I mentioned, from the output from this design of experiments, we can then develop a statistical model from this experimental data. You see the classic contour plot on the bottom chart on the left-hand side of the screen, that we're all familiar with this kind of representation. From that, a number of additional confirmatory experiments were done, and obviously based on this model, we can then develop a design space. And here the intent was to define a design space which achieved greater than 96% purity of the crude drug substance. And that's shown by the overlay plot on the right-hand side of the screen, where you can see the white region represents that combination of anti-solvent volume and temperature in this particular two-dimensional representation. That combination of those parameters will deliver greater than 96% purity of the crude drug substance. And the small region on the bottom right-hand of the chart, shown in reddish brown, obviously is a combination of parameters where you're outside of that desired quality. So everything very conventional. I'm sure you're all familiar with this kind of approach and representation. So then we got into discussion about, in the team, about the design space concept, and we had some really interesting questions, and I think it was Jean-Louis that asked a really fundamental question that caused a bit of head scratching amongst colleagues in Pfizer at any rate, which was, why did you want a design space? You know, kind of an obvious, simple question, but actually it's a very profound question, and it's profound because it has, it really makes you think about your strategy, not only for development, but for submission and subsequent operation in the commercial phase of the life cycle. And it led to a discussion, and in Pfizer, I think that the perspective was that a design space was almost a natural outcome of doing multivariate experimentation. If you'd done multivariate experimentation, you could develop a statistical model, you could get a design space. And I've been in conferences and heard other people talk, and it's pretty clear that some other companies have that impression. But I know that not every company approaches it that way. So it was a very profound, interesting discussion, and I would certainly encourage industry colleagues also to reflect on that kind of question. Because, you know, it may be that it's a specific regulatory approval objective. You need a design space because you've identified that you actually definitely need operational flexibility in a particular part of the process, perhaps for reasons to do with changes in operating conditions that you're anticipating as a result of changing the facilities, whatever it might be. So there could be something that you've identified during development that you know you're going to need that manufacturing flexibility. But again, our experience is that conversation can be very difficult between the development people and the operational people at the stage when you're doing these kind of experiments. It's not always easy to identify the flexibility that you might need. Hence, if you could get broad flexibility across the whole process, that kind of would act as an insurance almost. So it's an interesting conversation to have. One of the questions that we also considered was a series of questions that Evdokia Korakianiti, who's here somewhere to my right, working for the EMA, developed and provided to the case study teams to stimulate their discussions. And you'll see some of this output reflected more or less explicitly in the presentation today. One of Evdokia's questions was a very general question to the teams about whether what was discussed would have changed depending on the type of product or the dosage form or drug substance and so on. And certainly from the discussion we had within Pfizer and the case study team here, the general approach that I've just illustrated, we apply, as you've seen, to both drug substance and drug product, and that's true for also small molecules or the biopharmaceuticals that we're developing. So in principle, this general approach that's outlined in the ICH documents we felt could be applied in almost any circumstance. And you might derive different design spaces, but all fundamentally are still based on scientific understanding and a multivariate combination of the interactions of parameters and attributes per the Q8 definition. What I think we have seen both in Pfizer and certainly is reflected in the conversations that I've had with other colleagues in industry is that it would appear, not just here in Europe, but perhaps globally, that design spaces for more complex products, for example, biopharmaceuticals, as we'll see tomorrow in the case study from Novo Nordisk, may be harder to get approved because of the complexities and because of the uncertainties. And I'm sure we'll talk about that more both today and tomorrow. So from these discussions, I think there are already emerging a number of best practice recommendations that we can think about here. One of these is for industry, when should an applicant request approval of a design space? Does it make sense to do it at time of first approval? Or maybe it actually makes more business sense to wait until after approval? And again, that question and that answer could depend on which industry sector you're operating in. So the answer may be different for an innovator, or it could be different for a generic company, or it could be different for an OTC company if they're applying these principles. Applicants probably also need to think about the operational flexibility they need and the complexity of the product when considering whether to register design spaces as opposed to proven acceptable ranges. We've already seen from AstraZeneca this morning a reflection based on their experiences and some of the practical consequences of introducing design spaces into commercial manufacturing that's caused them to reflect on their strategies for development and in particular submission of requests for design space approval. And Pfizer has gone through the same kind of exercise where we've reflected on our experiences and have changed our consideration in terms of approaches to registering design spaces. One of the things that I think we've also discussed at great length in Pfizer, and we've reflected and we think will be a significant part of the conversation over today and tomorrow, is the role of the design space in the overall control strategy of the product. If you look at the definition of design space in Q8, it's very much about the assurance of quality being possible solely on the basis of control of the input parameters without needing to do any additional testing. And so clearly, if that's the approach that you're taking and that's the way the design space is playing that role within the control strategy, that may be different in terms of the risks associated with that compared to a design space where additional controls downstream perhaps or in addition to the design space are being applied. The risk is the risk of poor quality or detecting failures is clearly different in those two scenarios. And we'll talk a little bit more about that in this presentation, but also in the presentation that we'll see later on case study 5 this afternoon. So let's see how some of that thinking might play out in the next area for discussion, which is the scale-up and design space verification aspects of design space. I think we're all aware that design spaces are frequently developed using small-scale experimentation, and we're also all aware that scale-up effects can and do occur, and that we need to make sure that we understand those where they occur and that are under control, and that we don't have adverse impacts on product quality at commercial scale. So this has led to the debate that we've had industry and regulators over the last few years around design space verification. and the use of science and risk-based approaches. And that's really what the next few slides are about. This inevitably was reflected in the kind of questions we received during assessment. Now, one of the things that you'll be seeing over the case studies that we're talking about today and tomorrow is that inevitably these are not what products are being registered now or submitted now. They are products that were submitted a few years ago, and we've all learned from these experiences and things have moved on. But this reflects what we've seen at the time when these were done. And in this particular case, this product was submitted prior to the most recent guidance that we've seen around design space verification. And I think I'm right in also saying that it was submitted prior to some of the points to consider documents. So in some respects, it's totally understandable that one would get questions of the kind that we're seeing on the slide here. The number of sizes and batches employed in purge studies used to justify skip testing of palladium, the solvents and related impurities should be provided. And it was clear through our discussions that both Pfizer colleagues in the case study team and the regulators all agreed that it's important that we manage the impact of scale on manufacturing processes. And that is true, of course, whether we're talking about design spaces or proven acceptable ranges. You know, we need to understand that, and we need to assure the quality irrespective of the approach that we've taken in regulatory submissions. For this particular case, the purge understanding was actually developed using small-scale studies. What was done was worst case studies. So we would take the worst case output from a previous process step and feed that in terms of high levels of impurities into the next step to make sure that the chemistry was such that we had that under control in terms of the parameters, and that we were getting appropriate purges and elimination of the impurities. What was interesting, I think, was the discussion we had around the control of the palladium levels. And these were derived from small-scale experiments. And we talked about the scale-up effects and needing to assure this, and how predictable was it from the scale-up. And one of the thoughts or the approaches that we tried to use in Pfizer was to use this idea of thinking about the design space in combination with other controls. And we actually had a test control which was felt to be, from a Pfizer perspective at any rate, if you're doing a test, that's independent of the scale. It applies to whether you can do that test on small scale or large scale. It's a detectability element, and quality by design is about building quality and founded on understanding. And for drug substance, small molecule drug substance synthesis, there I think we have the best possibility because we're founded on first principles understanding of chemistry. So we have the best possibility of using that first principles knowledge in combination with experimental data to understand and use that science effectively in the design of our product manufacturing processes and control strategies. And it was interesting to talk about the fact that here we were not solely relying on the design space. We had an additional detectability factor. And maybe that then helped to reduce the risk associated with scale-up effects and would then feature into the thinking around the design space verification. And we'll see some of that, I think, in the next few slides. And as you can see, inevitably, at that stage, there was already discussion about how were we addressing the verification of the design space using appropriate approaches to think about this at commercial scale. And it led to us developing a design space verification protocol. And I think at least some of our thinking has probably helped influence the thinking in terms of the most recent regulatory guidance. So what was done at the time was to develop this design space verification protocol, which described the actions that we would take to confirm that the areas within the design space would consistently meet and maintain API quality. We talked a bit about how it would be documented and managed in the site change management system. And we agreed that for this, it would be filed in the regional section of Module 3, which is a European approach to dealing with this issue. We also agreed that the data generated from the protocol would not need to be submitted, but would obviously be inspectable. And the assessors noted that scale and equipment changes should not be in scope of this protocol because there was the legal requirement for variations to be filed if such changes would be met. So there's clearly a legal framework which we need to take account of. And I think sometimes maybe companies forget about that there are legal drivers to this. So we can't be quite as ambitious, I think, as we might like to be because the European Commission would ultimately get quite annoyed, I should think. At the time, it was interesting. There was a comment made about the design space verification protocol encompassing the spirit of process validation. And I think it's clear that while the two activities may have some similarities, what's very noticeable if you read the most recent guidance is that the guidance is quite careful to distinguish between design space verification and process validation. So they're not the same. There are important differences. And if you're engaged in this kind of activity, it's important to read that and understand that. Just looking at this, what we ended up submitting in a little bit more detail, the approach we were provided at the time, and I will remind you, this is 2 or 3 years old now, the initial verification was going to be focused on the normal operating ranges, and we were going to build off the data that we had from clinical stability and process validation batches. We talked about a general change management approach that we use in Pfizer, which I'm sure is similar to many other companies. So we would do a general science and risk-based assessment. If we were making a change, we listed out a whole series of kinds of considerations that would be thought about, and you can see some of them listed on the slide there, to help the assessor give a sense of how the change management system was working in the company. And then we also provided some more specific details on this particular product in terms of how we would manage post-approval changes to the process parameters, where we were moving from the normal operating ranges perhaps to areas of higher or unknown risk. So, for example, if we knew there was an edge of failure as one of the boundaries in a design space, and supposing you wanted to move towards that for some reason or other, clearly that would represent higher risk and might result in you needing to think a bit harder about the consequences of that and design an appropriate verification approach to deal and mitigate that risk. And that included selection of appropriate testing, where obviously you might do additional monitoring or testing as a result of that, and you might also do very specific testing. So not the routine testing that might be included in the specification, but something specifically designed to look for and detect risks that you might anticipate there being based on your product and process understanding. And at this point, I'm going to hand over to my colleague Tuna from the Norwegian agency, who's going to take you through the next series of slides and look at the current guidance versus the protocol and the information that was submitted at the time. So, Tuna.
Thank you. Recently, FDA and EMA published questions and answers on design space verification. This was not available at the time of assessment of the present application. On the other hand, as Graham mentioned, this application may have served as some of the basis for the development of the questions and answers. In the next 2 slides, items covered in this guidance are summarized in the left part, and to the right it is summarized how this was dealt with in the verification protocol for axitinib drug substance. As already mentioned by Graham, design space is often developed at lab scale. And the verification protocol should consider those parameters that may be scale dependent. On the other hand, if an applicant can demonstrate that the design space is scale independent, then a verification protocol is not requested in the dossier. Some principles are given in this document. Movement within design space may pose higher or unknown risks due to potential scale-up effects or model assumptions. It is not necessarily complete at the time of submission, and the verification should occur over product and process life cycle. It's possible to do initial verification at or near target operating ranges. And it's not necessary to repeat the small-scale experiments or not necessary to verify the entire design space or to identify the edge of failure for the process. And further, it should be guided by risk assessment, and it should consider the potential impact to product quality, and also the control strategy's ability to detect failures. And it should also be considered to do additional non-routine monitoring of quality attributes or process parameters. The submitted protocol for exitinib drug substance was in line with these principles. A design space verification protocol could include a list of scale-dependent parameters, a definition of the potential scale-up risks to the critical quality attributes, a discussion of whether the control strategy can address risks, and a description of any additional controls. In the submitted verification protocol, there was a long list of critical process parameters for the last 3 steps and also impacted critical quality attributes, together with the tests included in the control strategy and additional monitoring opportunities. On the other hand, the submitted protocol contained little information regarding definition of the potential scale-up risks to the critical quality attributes, and no discussion of whether the control strategy could address those risks. I will try to say something about the current expectations on specific information in the verification protocol. The protocol should be detailed enough to give an outline of the applicant's design space verification strategy at commercial scale. During our discussions for the preparation of this presentation, it was brought up by one of the Pfizer representatives that the level of detail should be less than for a post-approval change management protocol. We agree that the design space verification protocol needs less specific description on which movement in the design space would be relevant, and also less detailed information that what would be expected in a post-approval change management protocol. For movements in relevant directions in the given design space, it should be indicated the failure mode, the effect of failure, and the control strategy. For movements from normal operating range towards identified edge of failure, this could be a matter of concern, and this could require additional monitoring. A smaller movement from the normal operating range should be seen together with the control strategy in place. In the present case, palladium was controlled by a routine IPC test, and this could be taken into account. When deciding the need for additional monitoring, the magnitude of movement from the normal operating range and established interaction and the probability of failure should be considered. This is linked to process understanding. It should be described what additional monitoring is planned when given parameters are changed outside the normal operating range within the design space. Relevant monitoring and testing should be applied in order to detect the potential failure. Based on the risk, the right analytical technique should be used. The design space verification protocol submitted in 2012 was largely consistent with recent EMA and FDA guidance, except that it should have been more specific on definition of the potential scale-up risks to the critical quality attributes, and it should have contained a discussion on whether the control strategy could address the risks. A topic for further discussion tomorrow, if there's time, is to which extent is the potential risk from the development of the design space from only small-scale studies mitigated by the control strategy applied. We have now come to the third topic of this presentation, which is the presentation of the design space in the submission. During our case study discussions, it was asked if additional experiments were done to answer questions from the dossier review. It appeared that no additional experimental work was conducted to answer the questions. The data was already available. Based on this, we concluded that the presentation of the design space information was a critical factor in the review of the dossier and the questions asked. It may be a challenge where and especially how the design space information should be presented. In the description of the manufacturing process in sections S22 and P33, it should be clear which steps or unit operations are covered by design space. For a master batch record, on the other hand, this is a different issue. And as you know, the master batch record is not a part of the regulatory submission. It is a recipe, and probably from the description in a master batch record, it will not be possible to see if there is a design space involved. There is some existing guidance on presentation of a design space. There are given some examples for presentation in ICH Q8 and also in ICH Q11. You can see some of it to the right in this slide. And this has been further elaborated in the points to consider document, also shown to the right in this slide. Recently, a joint FDA and EMA document became available after parallel assessment of quality by design applications. It covers lessons learned. It is clear that there are different approaches in the USA versus Europe regarding expected description of manufacturing processes. In EU, there's a requirement for a full description of the manufacturing process in all cases. The inclusion of non-critical process parameters in manufacturing process descriptions may give a high post-approval change burden, and some may wish that this could have been handled in an easier way where design space has been established. It is a challenge to find a suitable level of detail to include in the description part of the documentation in S22 and P33. On the other hand, the descriptions should not be too detailed, and Graham will come back to this later in the presentation. In this figure, the gray region shows the optimal operating space for step 6 that gives the desired crystal form. If, for a master batch record, this region is to be described as a range, then a much smaller region within the quadrangle shown to the left will be described, resulting in a smaller operating range for the relevant parameters. In S22, a figure or a mathematical function may be used to describe the design space. This is a design space summary for one of the unit operations towards the end of the manufacturing process. It's taken from the development section of the file. It focuses on the critical process parameters. In this summary, as well as in the process description, the design space for the process parameters was expressed as ranges. From the start, non-critical process parameters were not included in the process description in S22. More guidance has become available after this assessment was done, and for description of the manufacturing process, it's expected that the manufacturing process description is adequately detailed and include all relevant parameters, both the critical and the non-critical. In this case, more details for non-critical process parameters were included during the assessment phase. The development work presented was mainly satisfactory, except that it would have been helpful to have more summaries on risk assessment. The presentation of the multivariate data analysis was also insufficient in some cases. In this application, it was some difference seen between the drug substance part and the drug product part. The documentation was better for the drug product than for the drug substance. In some cases, it was asked that the applicant should present statistical evidence to demonstrate that a certain variable or the interaction of variables was insignificant. A two-dimensional presentation of selected variables and their effects on a certain quality parameter was not considered sufficient to justify a design space. In order to further help companies to decide which information to include in the file, the EU PIT team has on the work plan to elaborate questions and answers on several quality by design related topics regarding level of detail in the dossier. Another experience from this case was that it's recommended to stick to the agreed ICH terminology. And I can also add that the companies should not make their own definitions of established terminology, or at least not include this in the documentation file. In this case, the use of the term key process parameter, which was intended to be something between a non-critical process parameter and a critical process parameter, was confusing and difficult to handle. During the assessment, the term key was changed to critical in the most critical documents in the file. I would then hand over to Graham again.
So thank you. So as you can see from this slide, even simple things like just making it very clear when you're asking for a design space and putting that in the process descriptions seems in retrospect blindingly obvious, but clearly wasn't at the time. And I don't think we're the only company to have done this either, as you'll see later. But so there are clearly learnings that came out of this in terms of presentation of information in the dossier. One of the things that Tuna mentioned was the tension that I think exists between having a sufficiently detailed process description, and as we've also already heard this morning from Frank and Olvia, the implications of that in terms of managing changes and deviations in the commercial phase of the product manufacture. But we still need to find a way of getting the value of this enhanced knowledge when it's presented in the dossier. And that relates to, I think, some questions about enhanced knowledge and appropriate commitments. And it's reflected in some of the questions that we had, as you can see. If a design space is being used, it should be obvious, it should be evident from the process description. But we were also asked that the design space include values for non-critical process parameters. And we've seen that that can lead to challenges and issues that perhaps still need to be discussed in terms of handling that post-approval, in terms of post-approval changes and deviations. It's clear that if you can do a good job in presenting the information and justifying why information is or isn't included, that it is possible to have that, to get it right. And there's an example of this where, at least initially, we had argued that process parameters for step 2 were not needed, and we had this question about information for parameters and values not included in the design of experiments being requested, but we were able to provide a response which satisfied the assessors in the sense of conveying the science behind this and why parameters at that step were non-critical because of the process knowledge and the science that underpinned the assessment of what we're doing. We didn't get it right first time, but we did get it right second time, I would argue. So there's hope for us, I guess. It's the conclusion. This leads really, I think, to some thoughts about potential best practice recommendations. It's clear that the regulatory process descriptions in S22 and P33 do need to be sufficiently detailed in order to give the assessor a good picture of the manufacturing process. It's clear they should include all the relevant process parameters that are linked to CQAs, and that you'll need to submit ranges or target values. It's also clear that there's a desire for the assessors to get that understanding that what industry would regard as non-critical process parameters also need to be included. But this does set up the tension that I've just talked about. It's probably also very helpful to have the descriptions in S22 and P33 explicitly state where a design space is being applied for, and also potentially the interactions, if they exist, between the parameters to be highlighted. And as we've heard from Tuna, a number of companies have used non-ICH terminology, and I think the difficulties we have with terminology means that it's a very strong message that don't use non-ICH terminology such as key, at least in submissions. I think it's okay in development or within a company, but when it comes to submission, try to stick to the ICH terminology. But this leaves us with some thoughts, I think, for topics for further discussion. We've seen this need for detailed process descriptions, but on the other hand, that has some consequences. If we include a lot of non-critical process parameters in design spaces or just in process descriptions anyway, it can lead to potential need for an increased classification in variations or additional variations that perhaps traditionally might not have been needed when process changes are required. I think we're all agreed that parameters don't affect CQAs equally. Some parameters have more effect than others. But when we were talking about possible solutions for this within the case study team, the conclusion that we came to was that maybe the regulatory framework as it is at the moment can't necessarily accommodate differences between critical process parameters and non-critical process parameters in process descriptions when it comes to thinking about post-approval changes. So I think that's something that we need to talk about because it's a fundamental issue. And what was interesting, I think, in some of the steering committee discussions, Keith Pugh, who is somewhere at the back, I think, had an interesting perspective that I'm hoping that he'll be able to bring forward and discuss with us at some point appropriately in the workshop over the next 2 days. I think the other thought, and you'll have got this sense already, is that in terms of our thinking about the focus that we've put on design space versus some of the other kind of QBD concepts, perhaps we've been focusing too much about design space. After all, design space is optional. You don't need to have a design space. In contrast, something like a control strategy is agreed to be essential. Every product has a control strategy. And you know, with a well-developed manufacturing process, it's how you deliver the quality that the product needs at the end of the day. So it does lead to this question again, which I think is worth further discussion about the role of a design space within a control strategy and whether thinking about that helps us with this question about the level of detail. So if you can stand back a little bit and look at the role of the design space within the overall control strategy, if it's having a less critical effect in terms of delivering product quality, maybe there isn't a need to worry so much about all the risk assessments or all the detail that underpins the experiment, and to focus on the right areas. Because at the end of the day, it's that assurance of quality and protection and delivery of public health, which is something that we all have a shared interest in. And of course, at the end of the day, we also need to think about this in terms of the regulatory framework in which we operate in terms of managing changes through the variations process. So with that thought, I'd like to finish and thank you very much for your attention and for Tona's excellent contribution, and open up the floor for questions.
Graham, we've seen in both case studies now sort of a strong push from European regulators for sort of this binary system of criticality. So it's becoming obvious. I'm curious to know from Pfizer's perspective how you've balanced that with more of the FDA's concept of continuum of criticality.
Yeah, that's a really interesting question, and I'll certainly ask my Pfizer colleagues to chip in if they've got interesting views as well. I think we all recognize, whether we're in Europe or US or Japan, that there is this continuum of criticality. It's how we actually deal with that's the challenge, because dealing with a continuum is actually pretty difficult, I think, when it comes to actually applying rules, if you like, within a legal context. At the end of the day, you have to decide whether something needs a variation or not, for example. And so maybe the question is not so much about criticality, but it's about post-approval change management and which category of variation something fits into or not. And you need that predictability. Although companies have concerns about this area, at the end of the day, I think what we also need is predictability. And having these rules and having a binary system, whether you agree with it or not, does provide some level of predictability. So it's clear that we're trying to wrestle with a number of competing demands, trying to reflect the real world and make that into something that actually works from a practical perspective. I'm not sure we have all the answers. I think it's a topic for discussion. Within Pfizer, we've recognized that the kind of 3 criticality level certainly is not something that's worked from a regulatory submission perspective. We've got the message, and we're using the binary system because that's been agreed through ICH, and it helps avoid some of the confusions that we are aware of. But I think internally we still recognize that there's a continuum, and we may still be using critical, key, non-critical internally within development to help the discussion about how to develop good quality processes and appropriate control strategies. John, I don't know if you want to say something.
Yes, I'm John Grosskopf, also from Pfizer, so I was just going to add to that. The question was about the contrast between how the EMA and the FDA has responded to the binary criticality format, and we actually have received very similar feedback from both agencies. In other words, that middle category that we've used historically, the key category, is not something that either region is very comfortable with as presented in a submission. To use that middle category as a development tool is something completely different. It's something that we still do in Pfizer. But when it does come down to a submission itself, we have to make a decision. It's either going to be critical or non-critical.
Graham, dare I add to that just a little bit more? I think the question says, is there a continuum of criticality? I think we all need to buy into the regulatory view that it's critical or non-critical. But I think what we've got is a continuum of residual risk. So once you understand the criticality of something, you can also understand your risk of failure. And that's where the criticality comes in. And then you can balance that against the change management requirements, the control requirements, the degree of detectability. Do you need to add controls to a design space that's working parametrically? So there's lots of things that an industry can work on around continuum of risk and how you manage that.
I'd like to talk about key for a minute. I don't think that key, from a small molecule perspective, I understand why key would not work. I'm Wendy Lambert. I'm back here.
Oh, sorry.
Hi. I understand in small molecules why the criticality continuum isn't crisp, why key works or does not work. But in large molecules, where we have impacted process performance as well as quality attributes, key works for us much better, and in some cases is needed, because after all, ICH Q10 points out that we should be monitoring our process performance in many places. So.
Yeah, thank you. Yes, if I can ask people to introduce themselves and maybe raise a hand. Christine, I can see you're wanting to say something.
Christine Moore, US FDA. I think this is an excellent discussion and some excellent points were made. Graham, I really like your comment that it's really about post-approval change management and how we deal with that. I do want to go back and talk a little bit about, at least on the small molecule side in ONDQA, how we've shifted our views on definitions of criticality. Before we moved into the EMA FDA pilot, we didn't question how people use terminology and using terms such as key, quality, process parameters. We saw multiple terminologies. And our philosophy at that point was, as long as the company clearly explains the terminology, we can evaluate that during the review. After several years and many different companies using different definitions, some for the same word like key, we realized, and in discussion with the EMA, that we had a communication and a knowledge management problem. And that when we had multiple companies using different definitions for the same term, for the same word, such as key process parameters, it became an issue with communication between ourselves and our other colleagues related to inspections. It became an issue with communication with our our pre and post approval. And after the discussions with the EMA, we decided that sticking to the ICH terminology, at least for the process description parts, is really best. And as was mentioned, it's up to each company to decide how you use terminology in development. But for clarity and continuity, we now agree, as we've stated in the Q&As, that we believe the ICH terminology is the best. Thank you.
Thank you, Christine. Gerd?
Yeah, this is, excuse me, Gerd Thurer from Roche Genentech. Very good discussion. I want to go in a slightly different direction maybe, and maybe it's a question we can park the answer for tomorrow, but I want to latch on to the fact that the concept of design space verification was really publicized in the Q&A document. And again, I'm not representing Roche's Genentech's opinion, but my personal curiosity. To me, the concept of design space verification is a new concept. It's not, the words don't appear in the ICH guidances. So to me, it is maybe a necessary solution, but a new concept. In the past, most interpretation of the guidelines or even new concepts have been somewhat consultatively or collaboratively maybe agreed on between industry and regulators, of course, in some sort of feedback mechanism. The ICH process, guidances with review periods, etc., public comment periods. It seems that there have been Q&As before, particularly by the EMA, as clarification. It seems that while it's a very effective process, the Q&A process to the joint pilot now lacks that element of reflection or industry input. And especially, I think, while some of the Q&A answers are very helpful and very clarifying, to me personally only, the concept of design verification is so new, additional. There was always a discussion about scale and scalability and scale factor, but it is a new element or a new interpretation, my personal opinion, and it is basically being introduced as an edict, but not in a way that we could have a reflection on that. So my question is more or less on the business process of the Q&A, whether that is foreseeable, the move forward to make further interpretation of QBD possible. Sorry, a long question. Maybe you can park the answer for tomorrow.
So if I've understood your question, I think it's probably more directed at regulators rather than myself as industry. So perhaps I can ask Jean-Louis or maybe Ed Doppia to comment.
I think, as far as I remember, I have to admit I have not the document in front of me, but for design space verification, this is in the reflection paper, although it points to consider a document. We have not given more detail how this verification should look like. This is correct. Regarding question and answer has some advantage that we have not to reopen and document the guideline and so on. So if we have to reopen the guidelines, this is from an administrative point of view much more difficult. For me, the design space verification is the same line as we have. This is a difference, for instance, with US, that we also ask for a process validation scheme. So in some occasion, if for non-standard, so-called non-standard processes, the process validation has to be already submitted at time of submission. However, if it is a standard process, and we are satisfied with the scheme. And this is a little in the same way as for the process, for the design space verification. So we do not ask for data, but we have to ask the company how they will make this verification. Having said that, when we discussed this with a company, it was quite interesting to see that also then the company started to think about how should this verification be, so that even at time of submissions they have not really made their mind how a verification should look like. So I think the discussion was very beneficial. And as far as I remember, but then I have to be corrected by Evdokia or by my other colleagues, but I think for this question and answer, we have also consulted. So we were no consultant, okay. This is maybe, but anyhow, once something is published, industry has always a possibility to comment. I don't know if Evdokia, you want to?
However, the output of this workshop will be used for the development of new Q&As and potentially revisit some existing ones if we feel the need that we can. So there is always an opportunity. What we felt it was extremely needed was at least to clarify what were the expectations at that point of time, because these were recurrent questions we were seeing in submissions. So it was like to say, this is the issue, this is how we see what we need, and yes, that's the purpose of this workshop, to hear the other views, and if needed, I would be reluctant to say go back, but if needed, why not? That's the purpose of this workshop.
Nana?
My name is Nana Kruse. I'm from the Danish Health and Medicines Authority. I'm assessing biotech products, and I think there might be some difference or some difference in expectation for the documentation that we need to bring into this, because if I have well understood, I don't know the chemical area or small molecule area, but if I have well understood the expectation with regard to batches, full-scale batches manufactured at the time of marketing authorization is different from biotech products where we have expectations of seeing at time of marketing authorization data generated with full-scale batches. So I think some of this might reflect that there is a difference in the requirement of data.
This is already reflected because there's an annex to the Q&A. Stepping back, hopefully what I would expect to come out of this workshop is that all these requirements, all these expectations are to be addressed on a case-by-case basis, also depending on the product, the complexity of the process, etc., etc. So everything is like, this is standard. However, if it's a very standard product, or if it's a very complex product, this could be adjusted. So the requirement, the level of the requirements could be adjusted depending on the product and the risk of the product and the risk of the design space, and etc., etc. So again, everything should be read into the context of what is in that dossier, in that submission.
Thank you. TG?
TG Venkateshwaran from Genentech. Just adding to the discussion on design space verification protocol, there's been a lot of talk about continual quality verification. Is this something that is envisioned to be part of that and will be rolled into that part of process validation? I was just kind of curious because this eventually would be part of process validation. So just wanted to throw that out there.
Ed Dochio, do you want to comment on that in terms of your thinking about the relationship between design space verification and continuous quality verification? Or process verification, continuous process verification?
Sorry, it was obvious I was not following the question. I'm really sorry for that. So if let me repeat to make sure I get it right. It's what is the relationship between design space verification and continuous process verification, correct? Continuous process verification maybe would address, is an alternative process validation, and process validation addresses the whole process, versus continuous process verification is specific to that step of the process that is covered from the design space, first of all. They could fit in, but they could be complementary, and as I said, these are personal views, they could be complementary, but one doesn't really replace the other. The purpose of design space verification is to address the scalability aspects. So there could be cases where the design space is scale independent, so there's no need for for a design space process verification.
I guess I can repeat my question there,
Okay.
okay. And my question was, do you see these being rolled into continual quality verification aspects of process validation?
Not really, to be honest. No.
There is a mix.
I think there was a question at the back. Yes.
I also have a question about design space.
Could you introduce yourself, please?
Oh, sorry, yeah.
Thank you.
My name is Åsa Herström Björling, and I'm from a Swedish agency. I'm sorry.
Thanks.
I have a question about slide 19, about design space verification on a larger scale, because I think the slide is a little bit confusing for me. In the dossier for this product, the company had results from laboratory and pilot scale, and then the company and the agency agreed on a design space verification protocol, and the assessor said that they don't need to see data, if nothing special. But then the last point says that the assessors noted that scale and equipment change should not be in scope. And then my question is, then you must mean scale exceeding the actual commercial scale, production scale, because otherwise I think it's confusing. Do you understand my point?
Yes, I think so. And I think, yeah, essentially there was what was clear, and John, you probably know better than I do. There were certain types of changes that would clearly fall within the scope of requiring variations according to the variations guidelines. Hence, you couldn't manage that kind of change through this verification protocol. John, do you want to add something?
Yeah, I was just going to say, your interpretation is correct. The variations, the type of scenario that would trigger a variation would have been outside of the registered range, right? So the verification protocol was independent of that. I guess what the assessor was really telling us was that the design space verification protocol could not be in contradiction with the variation guidance, so that there were specific changes in the variation guidance that still applied.
Good. Yes, Dora.
Just Dora Kurti from GSK. Just one question, actually clarification. When you started your talk, you made the statement, and I want to make sure you said that. You said that by definition, the design space, because it is a multivariate combination of raw material and process parameters and provides assurance of quality, it's almost as if leading to a control strategy where quality does not need to be tested. Did you say that?
Maybe, but not with your accent.
It's okay. The accent has nothing to do with the substance.
No, sure,
so,
that's right,
yes.
Dora. No, to be serious, what I think I was trying to say, and thank you for pulling me up on it, because I think it's an important clarification, was that, maybe if I put it this way, the way that we had used the design space concept in Pfizer was that we were not solely relying on a design space as defined in Q8 to provide control. There were additional controls that we were using. And our thinking around that concept is that because you've got that additional control, it reduces the risks associated, for example, with movement in a design space, and therefore could be helpful in terms of thinking about how you would approach design space verification or indeed other changes, because you've got additional detectability controls or whatever it might be. And so therefore, the fundamental message is don't look at design space in isolation, but look at it in context of the overall control strategy and how you're assuring the quality of that particular critical quality attribute or all the quality attributes that you're interested in.
Yeah, I just want to add a point here, and this comes from how every other industry sees the design space. The design space is the deterministic part. In other words, we define how I'm going to go from point A to point B. There is also the stochastic part, the unforeseen disturbances. And this is where every other industry puts the controls.
Okay.
So once we understand this, that we have to deal with two parts, then we define essentially our control strategy.
Yes, I think that's a very good comment. And I hope that that kind of conversation will get into that detail a little bit more in later presentations around the use of models and so on. So I think, yeah, let's hold that thought and come back to that, Dora, because it's a really important point.